Yes, I can. Should I start?
Yes, sir, you can start.
Okay. Thank you. Okay. So, as I was telling you that we need input data for our sequencing and everything from experimental science. So once we have isolated the nucleic acid, either total DNA or from intact cell or RNA or any kind of input data relevant according to our sequencing technology, we can do the genome sequencing, and based on those genome sequencing we can start the experimental, we can start the computational analysis of those experimental data, and I will tell you a little bit more about it in a while. So as I was telling you that or maybe I can just skip it. Okay, so once we have the, so as I told you earlier that there are two type of strategies. One is amplicon sequencing, which is just based on one single gene, and we say it like as I have written here, marker gene sequence reads. Okay, and the second is shotgun sequence read, which is metagenome, which is the total genome. Okay, so the pro-processing, as I told you, that quality is the major consideration when we do these kind of studies. So we do pre-processing, which include quality control and filtering the data based on our quality check. And once this step is done, we go to the next step where we try to identify similar OTUs, and then we try to identify their representative sequences. We do alignment and phylogenetic studies and find out in which find out their phylogenetic trees. Simultaneously, we also do the taxonomy assignment of those representative sequences and generate OTU tables. And based on those OTU tables, we do a lot of statistical evaluation and visualization using different analysis and try to come up with the relative abundances in different samples. Okay. In another part, when we have shotgun sequencing rates, or like the total genome sequencing of that particular sample, after again we will do quality control and filtering, but after this step we will do the assembly. Okay, so we will assemble the whole genome, and once we have assembled the whole genome, we do identify different genes, and based on different databases which which are there, we try to identify or characterize the function of those genes. And once we know the function of those genes, we try to cluster them or categorize them different functional tables. So in which kind of function they are involved, are they part of a pathway, or in which kind of pathways they are involved, in which kind of broad functions, let's say carbohydrate metabolism they are involved, or signal transduction they are involved. We try to categorize them based on those functions which have been predicted using different protein databases. Okay, so there are two types of this metagenome sequencing. One is 16S RNA and another is whole genome shotgun sequencing. So as I told you, 16S RNA is just one single gene based amplicon sequencing or metataxonomics. So as it is clear, because it is just based on one gene, and it is based on the whole genome of all the organisms which are present in that particular niche. So it will be cheap, obviously, because it is just one gene based, and it will be very expensive. Okay, it will give us relatively small amount of data, and it will give us a lot of data. It will provide genus level taxonomy. So again, if you are aware about taxonomy, then in taxonomy there are different lineage, different bullets, like kingdom, like what is the kingdom level of any particular organism, what is the class, what is phylum, what is order, or family, genus, species. So all these are different levels in taxonomy. So 16S RNA sequencing, it gives us only genus information. However, this one, shotgun sequencing, it will also give us information at species level or strain level. Okay, so it is quite depth. It doesn't have that much depth. Okay, and again, as I told you, it is reliable only to genus level, and we cannot identify functions here directly. Okay, and here we can identify precise functional inference for bacteria and archaea or any other microbes which are present there. Okay, so these two are the the basic ones, and these are their differences. And again, it depends on what is our question, and depending on that question, we can use any of these strategies. So let's start talking about the first one, which is 16S microbial community profiling. So how it works on the basis of 16S RNA gene sequence or any gene sequence. Again, if you are using some other gene, let's say a nitrogen fixation gene, NIFH, so it will become NIFH microbial community profiling. Okay, so and you can use, but most of the people use this sequence because it is highly conserved across all the bacterial organisms, and even after being conserved, it had some regions which are very diverse in all different taxonomy organisms. Okay, so there are these 9 variable regions which people can select and work with, and based on these 9 variable regions, people have determined different primers for different regions, and based on those areas, we can find out that we can do the genome sequencing of these areas, and based on those sequencing reads, we can find out that to which organism they have more closeness with. Okay, so this is a gold standard for bacterial diversity, and the pros is that it is ubiquitous, highly conserved, and evolutionarily stable. And the main negative aspect about 16S RNA is that in many organisms they are present in more than one copy. Okay, so sometimes it makes it a little bit tricky to use 16S microbial community profiling. Okay, but yeah, most of the people, when if you guys want to go ahead with 16S microbial profiling, most of the people, they prefer V3 V4 region, and it's like around 428 base pair, and we have standard primers for these different variable regions. Okay, so once we have, so let's say we have used V3 V4 region. So once we have this V3 V4 region, we will do the we will use those primers to amplify those segments, and then we will like do the sequencing so that we can get all the reads from that V3 V4 region from all the organisms which are present in that niche. Okay, once we have our reads, so reads are like a small fragment. So let's say if we are using V3 V4 primer, our reads will be around 400 base pair or 428 base pair. So based on these reads, so these reads will be, let's say if I can use any, I can use so reads are something like this. I will just quickly tell you here. So reads are like this. Okay, so we have like small reads. Okay, maybe millions of reads. So what we will do, we will try to identify. Let's say if we have 400 base pair segment. Sometimes we do 100 base pair sequencing. So first of all, we try to arrange them and make them a little bit bigger, so that they become like up to 400 base pair. And when we have those 400 base pair, we try to go to the next step. Sometimes if we just directly want to go ahead with these small reads, so we try to organize them in in we try to align them and see which are the representative ones for each of them. Okay, so that's how we do for all of these. Okay, another aspect here is once we have the data of all these reads, we will do first of all quality filtering, okay, or we will do pre-processing. So there are different tools which are available, let's say FastQC or Trigonometric or NGSQC toolkit, and based on these different tools, we try to do filtering, or we try to remove different not required sample, not required reads. So sometimes we add adapters in our sequencing, so we try to remove those adapters, something like that. Okay, so we keep only those reads which are of a good length, of high quality, and without any extra sequences or adapters. Okay, once we have the pre-processed data, After this, we try to divide reads into variable regions. So we try to align them all together, and then we separate them into forward and reverse, and then we bin reads into 6 regions based on start and stop codon. Okay, so we try to align all those reads with one each other and try to make their representative sequences. Okay, so and that's what we do here. We cluster reads into OTUs. So we cluster all the reads which are similar to each other into these OTUs, which are operational taxonomic units. And once we have these OTUs, we also have one representative sequence, and that representative sequence is used to assign taxonomy. Okay, so we compare it with different databases. That representative sequence, we compare it with databases and try to find out that in which organism it has maximum homology with. So we use different type of databases like Green Gene, Silva, NCBI, or MyRDP, and we try to find out that what is the taxonomy of that particular representative sequence. And the taxonomy of that particular representative sequence will be the same for all those reads which are clustered in that particular OTU. Okay, and then we compare those OTUs amongst different regions. Once we have all these abundance data, what we do is, okay, so it will be better explained here. So we have these 16S RNA reads. We try to amplify and sequence these 16S RNA. So let's say these are all different reads. Then we try to cluster them into similar ones. So this is one operational taxonomic unit. This is second one. This is third one. This is fourth. And then we use these different genes to identify the taxonomy of that particular OTU. Okay, and based on this analysis, we try to identify the abundance, and we also identify their taxonomy. Okay, we can also identify that which kind of variations are present within different sequences as shown here. Okay, so I don't know, like should I tell you this or but not, but let me see the time. So we still have time a little bit. So I will skip it, but this is just to tell you that when we do the sequencing, we get forward read and reverse read. And when we like their quality may be a little bit different. So this is a quality scale. So as you can see, this is a Fred score, and it determines that with, like, let's say the read is 150 base pair length. Okay, so it tells us that what is the quality of each base pair, and ideally it should be more than 20 at least. Okay, and if possible more than 30. Okay, so 20 Fred score means that there will be 1 in 100. Like if we sequence 100 base pair, then there will be one error. 30 means if we sequence 1000 base pairs or 1000 nucleotides, there will be one error. Okay, so based on that, we try to determine that how good the quality is. So this is the raw data. Okay, raw data. Now when we use like trigonometric, let's say if I have selected Fred score 30 and minimum length of 50 base pair, then it will remove everything which all the reads which has read read length less than 50 and Fred score less than 30. So I will get something like this. So if you see, if you compare the second figure with this second figure, so you will find out that all the reads which were of low quality, they have been removed here. Okay, and now we have very high quality reads. Okay, so once we have these reads, we will align them into OTUs, and we will do homology based taxonomy, and we will generate like these phylogenetic trees, and we will identify these kind of abundance tables. Okay, so once we have those abundance tables, we can use different creative methods like R, Circos, or ggplot to create figures and suggest that which kind of organisms are present there. So let's say this is the phylum level diversity. So it determines that firmicutes are present in like 50% around, and bacteroids are present in 40%, and actinobacteria are 10%, and proteobacteria are like 1% or something. Okay, and simultaneously we can do at different levels. Let's say class level, we can do the same analysis. We can do at order level, and we can go in as much depth as we can. Okay, so here this is genus level diversity, and we can find out that okay, bacteroids are present in the most, and later on we can also do analysis at strain level or species level depending on the methodology selected. Okay, so as you can see, these are just some representations from different studies. So this is phylum level diversity at different soil samples. Let's say this is desert, this is forest soil, this is grassland, this is arctic, and this is mangrove. So if you compare the desert one, in desert we have a lot of actinobacteria, but the abundance of this actinobacteria in other members is quite low. Okay, and if you see, they have proteobacteria here a lot, like around 30%, but here the proteobacteria has gone up to 70 or 90% in different organisms. So we can do all these type of comparatives and find out these kind of valuable information that which organisms are present there. Okay, and yeah, we can make all these different type of figures and try to analyze our data in a more sophisticated manner. Okay, we can also find out some different ecological concepts when we do these kind of studies. So we can find out, like, how rich that particular environment is, or how even that particular environment is. And means these are ecological aspects which we try to understand when we do these kind of studies. We can identify like alpha diversity. So what is the diversity present in a single sample, like one sample only. We can do beta diversity analysis, which will tell us, like, what is the diversity between samples. Okay, and then we can do gamma diversity, which will tell us, like, what is the total diversity in a landscape. Okay. Okay, so another aspect which I was telling you was the metagenomics, or the whole genome information, or the whole genome sequencing of microbiota. Okay, so for that we have another strategy, because now we have the whole genome, and the genome can be extremely large for some organism, and let's say 1 MB or 2 MB or 5 MB, okay, or maybe 10 MB. So we want to sequence the whole genome of those organisms, so that we can find out which genes are present, and we can we can find out their functions later on. So what we do for that is, once we have those reads, now we try to assemble them. So assembly is, as I was telling you earlier, is like this. So we have like these reads. Let's say we have these random reads. Okay, millions of reads, maybe of equal length. So what we try to do, we try to arrange them or find the overlaps between those reads. Okay, so we have different strategies, and we try to find out these kind of overlaps between two reads. Okay, and based on these overlaps, we try to generate one big contig. Okay, something like this. So there might be like these kind of several hundreds of contigs which we are able to identify. So the thing here is that, let's say there was a gap here. Okay, which means that no sequencing read was found in this region. So at that region they will create a break. Okay, and it will separate these two contigs. Okay, so we do these kind of comparisons, this kind of arrangement, and based on these arrangements we do the assembly. So once we have done the assembly, we we will map them to to known genomes, and we will try to identify specific genomes within that total genome. Okay, so we will try to identify bins. So we say them like genome bins. So using different softwares like MetaBat, we try to identify genome bins. So let's say in a metagenome there are 100 genomes, let's assume. So we try to identify those 100 genomes if possible, and then based on that we try to identify all the genomes, and we do genome annotation and identify annotation reports, identify their gene function, categorize them, and we also try to find out what is the taxonomy of each contig or each genome. We also try to identify what is the completeness level of that genome, or is there any heterogeneity in that genome present or not, or what is the assembly quality. So we do all these different analysis using different tools and try to get more information about our sample. Okay, I will not be going into this right now, but okay, so based on those abundance analysis, we can identify that in different samples which organisms are present. Okay, and as I was telling you that using metagenome data, we can identify information even up to strain level. So we have genus, we have species, we have strain. So strain is like quite in depth. So using metagenome data, we can even get information till strain level, and that is really helpful for many studies. Okay, so I will just quickly show. Okay, so once we have the whole genome, what it may tell us. Okay, so it can tell us about different functions or different metabolic pathways which we might be interested in, different secondary metabolites, and those secondary metabolite pathways, our housekeeping gene pathways, let's say glycolysis, Krebs cycle. We can identify all the proteins which are involved in those pathways or functions, and more. There is no limit. We can even try to do more sequencing and find out the relative breaks in between, and we can get like the whole genome, and find out every information possible, okay, about that particular niche. Okay, so what are the take home message from this is that and the main points which I want to pinpoint here is that nowadays science has become really interdisciplinary, and only by using experimental biology or any let's say physical sciences or soil sciences or any research area, uh, things are not covered as a whole. Okay, and to get a whole perspective, we need to have a balance or a juncture between different fields. So let's say if we are talking about soil sciences cannot be completed unless we are not including microbial analysis or identifying different microbes within those samples. Okay, because it may affect the soil architecture, soil salinity, or different behaviors, or not like as such behavior, but different aspects of soil. Okay, so the take home message is that like it or hate it, we cannot ignore the computational biology, at least in this present time. Okay, most of the people are trying to follow computational biology to get information about their research field. And like you may have a very like years long problem, and sometimes computational biology can have a quick solution. Okay, so but the good point is that obviously we cannot put experimental lab work aside. Okay, means computational biologists also need data, and that data will come from experimental biology only. Okay, again, computational science is to make your life easy, and again, you might have to use experimental science to confirm your results. But like let's say you have a very big data, or let's say if you have a soil sample, and you want to know which microbe is like kind of present in abundance, and if you are trying to do it using experimental biology, it will take let's say 2 3 4 months. But computational sciences, you can get it in 15 days or 20 days. Okay, so it will make your life easy. And mm, especially in microbiome field or metagenomics field, with current technologies, experimental science cannot give you information about uncultured organism. Okay, because there are a lot of organisms which cannot be cultured, but computational science can do it. Okay, because again, computational science is working based on ATGC. Okay, and again, people who are trying to do computational biology or learning computational biology, remember that there are limitations of computational biology. You always remember to play with cutoffs. Okay, there are different cutoffs, and you will get a different answer. Okay, and keep updating your databases are like the library for our research. Okay, and we cannot ignore them by not updating them. Okay, we should keep updating them, and always use multiple softwares before confirming anyone. And always remember that it is your mind that is giving order, not computers. Okay, so even if you are doing any computational biology or experimental science, you have to think about it, that what you are doing, why you are doing, and how you have to do it. Okay, so always remember all these aspects. Okay, and means if you want to take a snapshot of this, feel free to take it, because these are the three major papers which I really want everyone to read, who are or who want to go more into these kind of computational biology aspects, and they will really help you out a lot. Okay, and with this, I would like to just acknowledge IBAB for giving me a platform here. And it has been a wonderful time here from last 1.5 years. And I look forward to it. And also DST Inspired Department of Science and Technology for providing me DST Inspired Faculty Award. And yeah, these are my lab colleagues. So we have Vaishnavi and Shenu and Sneha. They are MSc Bioinformatics people, and they are really helping out in different lab projects. And I'm sure we will get some opportunity where you will listen to our work, which we have been doing. And these are MSc second year students who have been working with us, and some of them have worked with our SARS-CoV-2 analysis recently. So means in IBAB we have a very education plus research oriented environment. So we also involve our MSc students, our project interns in our research, so that they get an exposure of how to do the research in a better way. And we have project assistants, Animesh and Shraddha from outside, who are here for some research experience. And again, I just want to pinpoint that we are hiring. So if you know anyone who have MSc students who have their own fellowship, feel free to ask them to contact me, and we will definitely talk to them. Okay. And I really like this quote from Carl Woos, who was one of the pioneers in metagenomics. And he said that the time has come to replace the purely reductionist, eyes down molecular perspective with a new and genuinely holistic, eyes up view of the living world, one whose primary function is on evolution, emergence, and biology's innate complexity. And yeah, I mean, you should read this paper. It's a much read paper, and it really explains that how the computational biology might be the next solution of most of the things. Okay, yeah. So thank you, and I will be very happy to take any question now.
Hello.
Yes, sir.
Thank you very much, Dr. Gaurav, for your very nice elaborative presentation, which was very informative for us, especially for the students and the faculties. One thing just I would like to hear from you, that basically we are the soil scientists. Our study materials is soils, and you have already talked about the microbial community analysis, the
Mhm.
microbial community profiling, etc. So as you know that there is lot of diversity, it means microbial diversity within these soil samples. So through this metagenomic data set analysis that you have pointed out, if you want to analyze this different diversity, means different species of microbes that is lying within the soil, into the converting into the data set through these microbial
Mhm.
community profiling processes. So how to start with this initially?
Mhm. Yeah. So as I told you that the first step is always experimental, at least even for us. Okay. Uh, so if we have a soil sample, and if you want to know the microbial profiling, then you have to first of all, extract the DNA from that soil. Okay. Nowadays there are many direct kits available who can directly take the soil sample, and we can directly do the DNA isolation from them. Okay, it's a very easy process. The kits are available, and even manual methods are not that hard. Okay, so the first step is DNA isolation from those samples, and then we can decide that either we want to go for 6-ness profile, 6-ness and meta taxonomy, or the whole genome metagenomics.
Okay,
But the
thank you.
first step is always DNA isolation.
DNA, DNA isolation.
Yeah, yeah.
So from the student side, if there is any interactions, you have got a very good opportunity to interact with Dr. Gaurav. So you can ask questions if anybody from the student side. Hello. Anybody from the student side, please unmute. Is Saurav Kumar? Saurav Kumar, is it?
Saurav, if you have any question, you can ask.
Okay, nobody's there now. It's over to Kapudi. Please summarize the lecture in short, and then you have the formal vote of thanks. Okay.
Hello. Hello.
Yes, you are audible.
Yeah. So thank you, sir, for this wonderful lecture. And I'll summarize it for the students also. That you started with the phrase called microbes are everywhere. Then you moved on to the components of soil and very well explained the various definitions like microbiota, metagenome, metatranscriptome, and the holobiome concept. Followed by that, the biotic and abiotic factors which are responsible for this holobiome, which affect the holobiome. Moving on, you very well explained the comparative genomics and the databases like NCBI and how these databases are useful to convert a raw data into a meaningful data. Especially the questions like who is there and what are they doing. It actually differentiates the general 16S metagenomics and the functional metagenomics. Moving on, you have also explained the marker gene sequence and the shotgun sequencing approaches, and what are the pros and cons of 16S versus the whole genome sequence. Especially, you have given attention to the procedure, how the OTUs are generated, and how it is useful. Lastly, you have also explained a lot of software and the assembly statistics metafile. So I thank you, sir, for giving us such a wonderful lecture. Here in soil science, we are not equipped with so much of technology, but since these are just softwares, we will definitely like to learn it and then so that we can explore it in our own research. And since in Kuch Bihar, the soil is really acidic, and I'm sure that acidophiles and these kind of organisms will be abundant in these kind of soil. So thank you, sir, once again for this wonderful lecture. I'll transfer the mic to Shobik.
Thank you, Kapil.
Hello. On behalf of the organizing committee, I would like to thank the vote of thanks to our chief patron of the Dr. Chirantan Chattopadhyay, Vice Chancellor of this university, for facilitating the program. I would like to thank the patrons of this program, Professor Ashok Chaudhary, the Director of Research, Professor Somin Moitro, Dean Postgraduate Studies, Professor Dipindu Mukhopadhyay, Dean Faculty of Agriculture, Professor Pradut Kumar Pal, Dean Faculty of Horticulture, Dr. Rupak Sarkar, Dean Faculty of Technology, Professor Prabhat Kumar Pal, Director Extension Education, and Professor Bidhan Roy, the Dean Students Welfare. I would like to thank very specially to our HOD and the coordinator of this program, Professor Dipindu Mukhopadhyay, who also is Dean Faculty. And I would like to thank very much to our convener, Professor Abbas Kumar Sinha, joint convener, Dr. Ganesh Chandra Ponik, because they have done a lot to arrange this program. And my sincere thanks to all the teacher members, Dr. Amrit Tamang, Dr. Sovik Dev, Dr. Tapudev Karmakar, Dr. Tapas Kumar Pundit, and Dr. Hidayat Kamal Tarabdar for their continuous help. during organization of this departmental webinar series. My special thanks to Dr. Gaurav Sharma from the Institute of Bioinformatics and Applied Biotechnology, Bangalore. Sir, we are very honored to have you here. I actually not a very man of microbiology, but still then I listen to you throughout the lecture. I think in my research, I can have some, I cannot get everything you say because my domain is completely different, but I think I got at least somewhat from your lectures. And I work on soil carbon especially. So carbon is very much related to microbiology, and all the teachers, they are working in different aspects of soil. So I think we got enriched as well as the students. So sir, thank you very much.
Thank you so much.
And also I would like to thank to our students and all the other people who have listened to this program. They are part of this program. Without them, the program was for them. I would like to thank very much to coordinate everything and is still here to organize all the details. So with that, I'm handing over this. And if I forgot someone to mention, I'm sincerely sorry for that from the end of the organizing committee. So I am handing over the mic to our HOD, Professor Dibendra Mukhopadhyay.
Okay, thank you very much, Dr. Gaurav, and we'll be in touch with you in your future whenever possible. So
Sure.
now we conclude these sessions for this lecture series. Thank you all. Okay.